So hello students and welcome back again to another video tutorial by Genome Biotech. In this video tutorial, we would be talking about the process of intron splicing. So what is this intron splicing and how does the splicing take place? Which are the proteins which are being involved in the case of intron splicing? And in what manner the intron splicing takes place in an cell? So what do you mean by introns? Introns refers to the non-coding DNA which is present in majority of the eukaryotic genes. So the non-coding DNA which is present in majority of the eukaryotic gene is being constituted by the introns. These introns have to be sequentially excised off. Okay, such genes have a split structure in which segment of coding sequences called as exons are separated by non-coding sequences. There are coding sequences and coding sequences are called as exons, and these exons are separated by non-coding sequences. These non-coding sequences are also referred to as intervening sequences. These intervening sequences are also referred to as the introns. The entire gene is transcribed to yield a long RNA molecule, and the introns are being removed with the help of a process that is referred to as an intron splicing. Okay, so there is a process that is being removed useful for the removal of the introns, and that process is referred to as the splicing. Okay, so the introns are removed with the help of a specialized process. That process is called as splicing. So only exons are included in the case of mRNA. So in the case of mRNA, only exons are present, introns are completely being removed. The process which involves the removal or the excision of the introns and the ligations of the corresponding exons together is referred to as RNA splicing. Such type of process is commonly referred to as RNA splicing. You can just listen here. The process of excising the sequences in RNA that corresponds to introns and joining of sequences corresponding to exons is referred to as an RNA splicing procedure. So such type of procedure is commonly referred to as an RNA splicing. Mechanism of RNA splicing varies depending on the type of the introns. There are many types of the pre-mRNA introns. The two different types of the pre-mRNA introns are the GUAG introns, AUAC introns, and GUAG introns. And AUAC introns are being removed by a complex that is referred to as the spliceosome complex. Now, what is this spliceosome complex and how this spliceosome complex is being produced, that we have to see. Okay, so here what you find is that if this is the intronic sequence, see, this is an exon and this is the intronic sequence. So I'm just making a line. So this would be the intron. Okay, from here till here, there is an intron. Now, in the introns, there are certain conserved sequences which are present. Which are the conserved sequences in the introns? One is referred to as GU, another one is referred to as AG, while there is a conserved sequence which is present and that is referred to as the A. This adenine is commonly referred to as the branch point adenine. This branch point adenine has an hydroxyl group present over itself, via which it will attack the GUAG, will cause the cleavage reaction to take place, and ultimately the G will react with the adenine here, and here the reaction when will take place will lead to the removal of the intron molecule. A pyrimidine rich region called as polypyrimidine tract, which is not shown in the figure, is found in most cases. The branch point adenosine, the branch point adenine or the branch point adenosine is the most important nucleotide that takes place in the process of splicing, which is usually 20 to 50 base pair in the length, and from the 3' splice site it is present. The AUAG and GUAG introns are the two different types of the intron. GUAG intron is one type of an intron. Okay, so there are two different types of the introns that you can describe: GUAG introns, and another one is referred to as AUAC introns. AUAC introns. So these are the two different types of introns which participate in the process of splicing. Okay, and they involve the process of splicing. This is the splicing process that is being involved. Okay. Now, how does splicing take place? In what manner the splicing takes place? There is a transesterification reaction that is responsible for the splicing to take place. In the transesterification reaction, what you find is that there is a branch point hydroxy group which is present. This adenine is commonly referred to as the branch point adenine. This adenine has a hydroxy group present over itself. The hydroxy group which is present over this adenine is referred to as the branch point adenine hydroxy group. This hydroxy group will attack the phosphate molecule. It would be attacking a particular phosphate molecule. Once it would be attacking a phosphate molecule, it will lead to the formation of the first transesterification reaction in which the first lariat-like structure will be produced. A lariat or a loop-shaped structure will be produced. So here there is a loop-shaped structure. Okay, this is a loop-shaped structure. Loop structure. Okay, so this is a loop structure which is present. Once the looped structure is being present here, then what will happen? There would be a second transesterification reaction. In the second transesterification reaction, there is the exon 1 which is present. This is exon 1. Sorry. There is an exon 1 that is being involved, and this exon 1 has an hydroxy group. This is exon 1. This is exon 1. Exon 1 has an hydroxy group, and via this hydroxy group, the oxygen atom will attack the innermost phosphate. When it will attack the innermost phosphate, it will cause the release to take place, and there would be a lariat like structure that would be released, and the exon will join with the exon number 2. Exon 1 will be joining with the exon number 2. This is exon number 2. Exon 2. Okay, so exon 1 and exon 2 are going to be ligated. So this is how the exon 1 and exon 2 are going to be ligated. And ultimately, what you find here is that the intron is excised. This is intron excised. This is intron that is excised. And this is how exons are being joined. Exon 1 and exon 2. Okay. So this is exon 1 and exon 2 that have been ligated and the introns have been excised off. Now let us look at how the entire reaction takes place. What are self-splicing introns? The group 1 and the group 2 introns. Firstly, I will be talking about the group second introns, that how does group second introns work? How does group 1, group second introns work? What is GUAG? What is AUAC introns? And then we would be looking at another type of splicing involving the splicing apparatus. So without wasting any time, let us move at the animation, okay, where you will get the animated version of the transesterification reaction and the process of splicing. So let us begin with the group second intron splicing. Okay, so we are going to talk firstly about the group second intron splicing, that how does group second intron splicing takes place and what is the spliceosome complex and how this spliceosome complex is being produced. Okay, before moving to this particular complex, let me give you a guideline, a quick revision of this a quick overview of this particular topic that we would be talking about here. Majority of the eukaryotic RNA contain stretches of non-coding sequences, introns. The introns are being surrounded by the coding sequences called as exons, and the introns have to be specifically removed. Now, how the introns removal takes place, that we would be visualizing. So please do view this.
Almost all bacterial RNA molecules are made up of contiguous stretches of codons, in which the codon for each amino acid is immediately adjacent to the codon for the next amino acid in the polypeptide chain. However, most eukaryotic RNA molecules contain stretches of non-coding sequence called introns. The introns are surrounded by coding sequences called exons.
So there are the introns and exons which are present. Introns are the non-coding sequences, whereas exons are the coding sequences as it is very clearly shown. So what is going to happen is that the introns are going to be removed and the exons are going to be ligated together.
Before these RNA molecules can be used in the cell, the introns must be removed and the exons must be spliced together.
So here, what has to be done is that the introns have to be specifically removed and the exons are to be ligated together. Now, how the introns are being removed and how the exons are being ligated together, that we will have to visualize.
While most nuclear pre-mRNA transcripts are spliced using a large complex called the spliceosome, some introns are capable of catalyzing the chemistry of their own release. These self-splicing introns are divided into two classes based on their mechanism of self-splicing: Group 1 introns and Group 2 introns. Group 1 introns are described in a separate exercise. When you have completed this exercise, you should understand the mechanism of splicing by Group 2 introns and the spliceosome.
Now, what you have to understand, you must know what are the group second introns and the spliceosome apparatus. How does group 1 and the group second introns work and what are these introns? Firstly, we must understand the mechanism of splicing by group second introns and the spliceosome complex. So what is the group second introns and how does group second and group 1 introns catalyzes the process of splicing, that we will have to visualize. So group 1 introns, you can find that group 1 introns will have a completely different structure as compared to the group second introns. The structures that we have already seen are conserved residues present in the case of group second introns. They can be GUAG or it could be AUAC. Okay, so having a look at the group second introns, and these group second introns can be catalyzing their own removal, then they would be referred to as self-splicing introns, and sometimes they will require a protein complex that would be referred to as the spliceosome protein complex that would be catalyzing its removal. Have a look.
And understand how the spliceosome directs RNA splicing.
So we must understand how the spliceosome direct the RNA splicing, in what manner splicing takes place.
Both Group 2 self-splicing introns and introns spliced by the spliceosome contain an adenine at a site within the intron called the branch point site.
So
These introns also contain a conserved guanine at the 5' end of the intron in a region called the 5' splice site, and another conserved guanine at the 3' end of the intron in a region called the 3' splice site.
Now please look here. I have already told you that the group second introns that are being shown to you here can be of the following two different types. They are basically the group second introns can be classified into the following two different types. Okay, group second introns. How they could be classified? They could be classified on the basis of the conserved conserved nucleotide GUAG, GUAG, and AUAC. So these could be GUAG, AG introns, and AUAC introns. So these are the two introns, two different types of introns that could be taking place. GUAG and AUAC introns. These are the introns that could be present because they have the conserved guanine and uracil residue at the 5' splice site, or has a adenine and a guanine residue at the 3' splice site. There is a branch point adenine, and this branch point adenine will contain a hydroxy group present over its structure. When this hydroxy group will be present, this hydroxy group will make an attack at the guanine, forming a loop-like structure that would be separated in the form of a lariat. Now, how does the reaction takes place? Let us have a view.
In the first of two reactions, the 2' hydroxyl of the conserved branch point adenine acts as a nucleophile and attacks the phosphoryl group of the conserved guanine in the 5' splice site.
Okay, so you can look here. There is the first transesterification reaction in which the branch point adenine hydroxy group is going to react with the guanine residue at the 5' splice site, forming a first lariat-like structure. Please have a look again.
Reaction is a transesterification, in which the phosphodiester linkage at the 5' splice site is replaced with a new phosphodiester linkage between the branch point adenine and the 5' splice site guanine. This three-way junction is the reason the adenine is called a branch point.
Because of this three-way junction, the adenine is commonly referred to as the branch point adenine in nature. Next.
In the second reaction, the newly liberated 3' hydroxyl of the 5' exon acts as a nucleophile and attacks the phosphoryl group at the 3' splice site. This transesterification reaction joins the 5' and 3' exons and liberates the intron. The
So
liberated intron is in the shape of a lariat.
you can look here. The next reaction takes place when the 3' when the exons see when the 5' exon attacks a phosphoryl group at the 3' splice site. The 5' splice site is being made free because the formation of a loop shaped structure when the guanine at the 5' splice site have been bounded with the branch point adenine. The 5' splice site will attack the 3' splice site and will lead to the joining of the two exons together with each other, and the intron has been removed from the process. Okay.
For almost all introns, splicing is carried out by a huge complex called the spliceosome. This complex is composed of 150 proteins and 5 RNA molecules called small nuclear RNA. Each of these RNA molecules is complexed with several proteins to form complexes called small nuclear ribonuclear proteins, or SNRPs.
Now, this is important. Most spliceosome is composed of more than 150 different types of proteins, and those proteins are snRNPs, which are also referred to as small nuclear ribonucleoprotein, and RNA-free proteins, which are present. Now, these snRNP and RNA-free proteins bind together with each other in order to form more than 150 proteins, and there are 5 RNA molecules, and those 5 RNA molecules are commonly referred to as snRNA. snRNA refers to small nuclear ribonucleic nucleic acid. So snRNA are going to work, and these are the different types of snRNA that you can find. They will constitute the complex spliceosome, okay, and then will catalyze the process of splicing.
Introns spliced by the spliceosome contain conserved sequences at the 5' splice site and 3' splice site, in addition to the bases directly involved in the splicing reactions. The consensus sequences shown here are for humans. The most highly conserved sequences are a GU in the 5' splice site, an AG in the 3' splice site, and the A at the branch site.
So you can find here that whenever the spliceosome complex is going to work, the spliceosome complex is going to catalyze the process of splicing. It is going to catalyze the process of splicing by catalyzing the removal of, or by catalyzing the attack of the hydroxy group of the adenine over the guanine residue, leading to the formation of a loop or lariat like structure. It will lead to the formation of a loop, or it will lead to the formation of a lariat-like structure. Now, how this loop or lariat-like structure will be produced? This would be produced when the branch point adenine hydroxy group will react with the guanine molecule, and the first transesterification reaction is going to take place. As the first transesterification reaction will take place, then it will lead to the formation of a loop-shaped structure. So this is the 5' splice site. This is the 3' splice site that is present. Now this process will be mediated by all these different types of proteins. The same diagram is also being represented in your Pathfinder. Please do have a look. This all has been discussed and told to you that how the lariat like a structure is being produced. Now what we would be talking about is the splicing apparatus or the spliceosome complex, that how does the spliceosome complex works. How does GUAG introns are being removed with the help of specialized protein and RNA complexes called as snRNAs, which are also referred to as small nuclear ribonucleoproteins. Okay, so these are referred to as short RNA molecules which are being associated with the specialized proteins, and those proteins are called as small nuclear ribonuclear proteins, also referred to as snRNPs. So these snRNPs are going to work, and these are the snRNA U1, U2, U3, U4, U5, and U6 are the snRNA which are present. The complex which is being produced is referred to as the spliceosome complex. There is the complex which is being produced, and this complex is called as the spliceosome complex. Okay, now how does spliceosome complex works, and what is the function of all these different types of snRNPs that we will have to visualize. So looking at the animation first, it is going on. Okay, so starting again from the animation, that how this process takes place, have a look.
First, the U1 SNRP recognizes the 5' splice site using base pairing between its RNA component and the RNA transcript. A subunit of an RNA-free protein called U2 auxiliary factor binds to the conserved pyrimidine tract, and another subunit of U2AF binds to the end of the 3' splice site. U2AF recruits branch point binding protein and helps it bind to the branch site. This arrangement is called the early complex.
The first complex that is being produced, which is referred to as the early complex, as you can find, is that the U1 is being bound to the 5' splice site. The branch point binding protein is binding to a branch point, and that branch point is the adenine. The third protein that is taking part is referred to as U2AF, which is causing the branch point binding protein to recognize the 3' splice site. And such type of complex which is being produced is commonly referred to as the early complex. Have a view at the pathfinder. Now what you are finding here is that what you are finding here, U1 BBP and U2AF. Okay, U1 BBP and U2AF. Now see this, U1 is going to bind to the 5' splice site. The branch point adenine is being recognized by the BBP. The branch point binding protein is going to recognize the branch point, and ultimately the U2AF is going to bind. Now the branch point binding protein will be released, and the U2 snRNP will bind, that will move towards the U1, leading to the formation of first lariat like structure. Okay, already shown in pathfinder. So this complex was referred to as the E complex. E here refers to the early complex which is being produced. This is commonly referred to as the early complex. Okay, this is the early complex which is being produced. Okay, let us have a visualization. Just a second. So this is the early complex. This is the early complex which has been produced. Now moving forward.
The U2 SNRP then replaces BBP at the branch site, aided by U2AF. This is called the A complex.
The next complex.
The branch site A remains unpaired and forms a bulge.
This complex is commonly referred to as the A complex. In this complex, the branch point adenine forms a bulge-shaped structure. You can have a look that this is the branch point adenine. When this U2 is going to bind, it will lead to the formation of a bulge-shaped structure, and this bulge-shaped structure will allow its hydroxy group to be exposed, and this hydroxy group reaction has to take place to this 5' splice site. Okay, and it will have the conserved residue GUAG. But for this process, the two splice site, the 5' splice site, must come in close proximity with the adenine hydroxy group. For this, the complex, the U1 protein and the U2 protein now began to move towards each other, causing the formation of a loop shaped structure. Okay, loop structure. Okay, now please have a look. So looking forward.
U2AF leaves the complex. The complex rearranges to bring all 3 splice sites together. The U4, U5, and U6 SNRPs join the complex, converting it to the B complex.
Now, the next complex is being produced. That is, all the SNRNPs are now being joined, and it will lead to the formation of a complex that is referred to as the B complex. So here you find the A complex and the B1 complex. Ultimately, U1 will be released, U4 will be released, and then the reaction will take place. Moving forward.
U1 then leaves the complex, and U6 replaces it at the 5' splice site. U4 is released from the complex, allowing U6 to interact with U2, producing the active site. The active site is believed to be comprised solely of the U2 and U6 RNA molecules. The branch site A attacks the 5' splice site, forming the three-way junction. Note, in reality, the branch site A and 5' splice site are much closer together for this reaction.
Now, very important point is that the active site is always made up of U2 and U6. Please do read it once more. U4 is firstly being released from the complex, and once the U4 is being released, then it is the U6 that comes and interact with the U2. Once the U6 has bonded with the U2, then it will constitute the active site of the enzyme. The active site is believed to be comprised solely of the U2 and U6 RNA. A branch site A attacks the 5' splice site, forming a three-way junction. There is a three-way junction that is being produced. Note, in reality, the branch point adenine and the 5' splice site are much more closer to each other. Next.
U5 helps bring the 5' and 3' splice sites together, facilitating the second transesterification reaction. The spliceosome then releases the mRNA product and the intron lariat. Note, in reality, the 5' and 3' splice sites are much closer together for this reaction.
Now the second set of reactions takes place. In the second set of reaction, the U5 helps to bring the 5' splice site and the 3' splice site closer together with each other. 5' splice site, which was having a guanine, will be attacking the 3' splice site and will lead to the formation of a lariat-like structure that would be removed.
U5 helps bring the 5' and 3'
helps to bring the 5' and the 3' reaction together with
prime splice sites together, facilitating the second transesterification reaction. The spliceosome then releases the mRNA product and the intron lariat. Note, in reality, the 5' and 3' splice sites are much closer together for this reaction.
So this is how the reaction was taking place. Next.
How well do you understand the spliceosome? In this section, you will find out
that how you are going to understand the spliceosome complex, that you will have to look at this particular section.
Question 1. Use the SNRPs and RNA-free proteins to assemble a spliceosome on this RNA molecule.
Now, see this. U1 is going to bind to the 5' splice site. Okay, so
correct.
U1 at the 5' splice site. Then branch point binding protein will always bind to a branch point.
Incorrect. Okay. You may want to
please. Branch point binding protein will be binding to this particular branch point. Now, how to make it bind? I should place it here. I should place it here. I should place it here. Where should
incorrect.
I'm going to place it somewhere now. It has to branch point binding protein has to be placed and will have to recognize this branch point.
Incorrect. You may want to.
This is how the
correct.
Okay. Now why it is not being showing? Now it will show.
Correct.
Okay. See, because firstly the 5' and the 3' splice sites are being bounded, then only the binding takes place to the branch point adenine. That is why it was not allowing me moving forward. Okay. Now you have not completed. Okay. So return to the question. Now. Because this must proceed now.
Incorrect.
Then only I can bind all the different types of proteins.
Incorrect.
Oh, sorry. Return to question. Then what I must do? This will bind only when the branch point.
Correct.
Okay. This is saying that when you will bind it, then only the process will be continued. Now, Once it has been left, then this complex will be bonding to this particular. Now the reaction.
Incorrect. You may want to try.
The reaction will proceed, but how to proceed the reaction?
Incorrect. You may want to try.
Just a second.
Incorrect.
Now this will say. Okay, this would be binding to this particular position so that the lariat-like structure could be produced. Then the U4 has to be removed. Now,
Incorrect. Incorrect.
just give me a minute so that at least I can show it to you that how this would be done, because I'm unable to remove all these. And where will I should take all these molecules so that the binding should take place? Because
Incorrect.
in this particular question, what we have to answer from here would be that here you will have to remove the U2AF protein. U2AF protein has been removed. Now you must remove the U1 protein for the next trace step to take place. Okay, so we would be removing the U1 protein. Okay,
Correct.
next, after the removal of the U1 protein, you can find that the next protein that has to be removed in a sequential manner would be the U4 protein, so that the U6 can interact with the 5' splice site. So the next protein that has to be removed has to be U4.
Correct.
Okay. Now the U6 will bind to the 5' splice site. The reaction will take place. And once the reaction will take place, it will lead to the formation of a loop-shaped structure. This is the structure that has been produced, and the removal of the molecules have taken place in the form of a lariat shaped structure. Moving forward.
Question 2. SR proteins bind to sequences called exonic splicing enhancers and recruit splicing components to the 5' and 3' splice sites. What would happen if SR proteins recruited the U1 SNRP and U2AF to the sites shown?
Okay, so if this SR protein is going to uh, upload the U1 and the U2AF at the present sites shown, then the exon 2 is likely to be excised off. The spliceosome would not be able to assemble. The spliceosome would assemble, but the RNA transcript would not be spliced. Okay, where is the branch point adenine? There is no branch point adenine. The transcript would be spliced joining the exon 1 and exon 2, exon 1 and exon 2. Okay, and the third would be RNA transcript would be spliced using exon 1 and exon 3. So exon 1 and exon 3 are going to work together. What would happen if the SR protein recruit the U1 SNRNP U2F at the site shown in the figure? So these are the sites, but there is no branch point adenine. So most
Incorrect.
the spliceosome would not be able to assemble. The spliceosome would assembly, but the RNA transcript would not be spliced. Maybe this.
Incorrect.
Okay. Exon 2 and exon 3. No, exon 1 and exon 3.
Correct.
Okay, how does this takes place? See, SR proteins helps to ensure that the spliceosome binds to true splice site. SR proteins can also be used to splice the RNA transcript in different ways. Okay, alternate splicing can be catalyzed by another class of proteins, and those proteins are called as SR proteins. Okay, so these will be catalyzing the process in different ways, allowing the eukaryotes to produce multiple proteins from the same primary RNA transcript.
Group 2 self-splicing introns and introns spliced by the spliceosome share a common mechanism of splicing. In the first reaction, the 2' hydroxyl of the branch point adenine attacks the phosphoryl group of a conserved guanine in the 5' splice site, forming a three-way junction.
Okay, this is the three-way junction which is being formed when the branch point adenine hydroxy group has attached to the 5' splice site.
In the second reaction, the 3' hydroxyl of the 5' exon attacks a phosphoryl group at the 3' splice site. This transesterification reaction joins the 5' and 3' exons and liberates the intron in the form of a lariat.
Okay, this is it. Once it has been removed, then this is the conclusion that we have obtained from the group second introns.
For almost all introns, splicing is carried out by the spliceosome. The spliceosome is made up of small nuclear ribonuclear proteins, called SNRPs, as well as RNA-free proteins. A complicated and precise series of interactions ensures that the RNA transcript is processed properly.
This is how RNA molecule will be processed. The U6 will bind, yet the U6 binding will ensure the attack to take place. Once the attack has taken place, it is the U5 that will catalyze the second attack to take place, and then joining the two exons together with each other, and the intron is being excised off.
You have completed this exercise.
Okay, thank you. Now we must look at Pathfinder, that what does Pathfinder says in the same process. The same process in the case of Pathfinder begins with the commitment step. What do you mean by commitment step? The commitment complex, also referred to as the E complex, the first complex, initiate a splicing activity. It is a complex that will initiate the first step of the splicing procedure, that is referred to as the splicing activity. This complex comprises of an snRNP, which is referred to as the U1 snRNP, which binds to the 5' splice site. Okay, partly by an RNA base pairing which is present. Okay, then the splice site is being bounded by a class or a member of family of proteins, and those are referred to as the SR protein that catalyzes the process of splicing. The name SR protein is given due to the presence of arginine and serine, which are rich amino acid sequences present in this protein. Binding of U1SNRNP to the 5' splice site is the first step in splicing. Once the U1SNRNP has been bounded to this particular site, that is referred to as the 5' splice site. Splice site is the first process in which the splicing should take place. Okay, here from the splicing will occur, and here from what will happen, the branch point binding protein which has been binded to the adenine residue and the U2AF which is present, the U2 protein will take the spliceosome protein snRNP will take the place of BBP, branch point binding protein, and the U2 will move towards the U1, leading to the formation of the second complex which is commonly referred to as the A complex. Look here, the pre-spliceosome complex. The pre-spliceosome complex, which is referred to as the A complex, comprises the commitment step plus U2 snRNPs, the later attached to the branch point. At this stage, an association between the U1 snRNP and U2 snRNP brings the 5' splice site in close proximity with the branch point. Okay, so there is the U1 splice site, U1 snRNP, there is the U2 snRNP, and the U1 and U2 snRNP are being made to move towards each other, and they are being brought in close proximity to each other. The spliceosome complex is being produced, is being formed when U6 and U4 snRNPs and U5 snRNP attach to the pre-spliceosomal complex. Okay, so these three molecules attach to the pre-spliceosomal complex and then would be leading to the formation of the spliceosome. So these are the three snRNPs which are present. These are the three snRNPs which are present, small nuclear ribonucleoproteins, and they will be assembling together, leading to the formation of a complex spliceosome apparatus. This is the complex spliceosome apparatus. Spliceosome some apparatus. This is the complex spliceosome apparatus which is being produced. Ultimately, what will happen? The U1 component will be released. Look here, the B1 complex is formed when the U5 and U4 U6 snRNP binds to the A complex. This complex is regulated by another class of proteins, and that protein would be referred to as the spliceosome protein, since it contains the component needed for the splicing reaction to take place, as it contains the component that are necessary for the splicing reaction to take place. It is converted to the B2 complex after U1 is being released. So it is converted to the next complex that is referred to as the B2 complex. The B1 complex gets converted into the B2 complex. Okay. The catalytic reaction triggers by the release of the U4. This require hydrolysis of ATP. Then the U4 is being released. This is mediated by the net release of the ATP, the hydrolysis of ATP, which will be converted into adenosine diphosphate and inorganic phosphate that would be released. Next, the U4 dissociate from U6 snRNP. Once the U4 dissociate from the U6 snRNP, this will allow the U6 snRNP to get base pair with the U2 snRNP. The U6 snRNP will mediate the first attack of the branch point adenine to take place. Okay, this would be the first attack mediated by U6. Once it will mediate the attack to take place, then U6 will attack, and once it will attack, then it will catalyze the attack to take place. Hydroxy group will attack at this particular point, will cause the release of this lariat like structure. Next U5 will act, next U5 will act, and U5 will catalyze the next release to take place. Here the hydroxy group which has been produced should make an attack over the 3' splice site, 3' splice site. Once the attack takes place, the process involves the removal of the intron. This is the intronic sequence removed. Sequence removed. Okay, this is the intronic sequence which is removed. Any doubt? Any problem? Okay, so this is how this splicing takes place. In the case of alternative splicing is a type of splicing in which the same gene or the same pre-mRNA can be processed in more than one form. How the same pre-mRNA can be processed? The same pre-mRNA consists of different introns and exonic sequences. This is the intron that is present. Okay, this is the intron that is present, and then there are the exonic sequences which are also present. This is the intron. Now introns could be processed in more than one manner with the help of proteins called as snRNPs. Okay, see this, these are the proteins which are involved, SR proteins. First, for example, if SR proteins are going to bind differentially, then they will catalyze alternate splicing catalyzed by the differential catalyzed by the differential binding of SR protein, by the differential binding of SR protein. Of SR protein. So SR protein is being differentially binded, and then it will lead to the alternate splicing to take place. It will lead to alternative splicing. Okay. Next, alternate splicing has been documented in many different eukaryotic genes, especially in the case of Drosophila melanogaster. The alternative splicing has been seen. Now, how does alternative splicing takes place in Drosophila? In Drosophila, this single pre-mRNA is would be processed into more than two different types of mRNA, in which the exons will be excised off along with the intron. In the above case, you can see that the exon number 3, exon number 4 has been excised off. In the above case, I'm just writing it out. Exon 4 has been excised off. Exon 4 has been excised off. Has been excised off. Okay, so there is the exon 4 which has been excised off. Here, what you can find in the diagram below, exon 3 has been excised off. Here, the exon number 3 has been excised. So this is how the alternative splicing is taking place. So in the case of Drosophila melanogaster, the alternative splicing takes place by the in the genes which are referred to as the Sxl gene. The three genes which involve alternative splicing are the Sxl gene, which is referred to as sex lethal gene, the transformer gene, also referred to as Tra gene, and the double sex gene, which is also referred to as the Dsx gene. These three genes involved alternative splicing. Now, in what manner the alternative splicing takes place, that we will have to visualize. Just a second. Now, the sex in the case of Drosophila is being determined by the genic balance theory. You know all that sex in Drosophila is determined by the genic balance hypothesis. Genic balance hypothesis. This is how the sex in the case of Drosophila is being determined. That means sex index ratio is being calculated. Sex index ratio. Now, sex index ratio equals to number of X chromosome divided by number of set of autosomes. Number of X chromosomes divided by number of set of autosomes. Divided by number of autosomal sets. Somal sets. Okay. So we all know that the sex index ratio in the case of a Drosophila, where the number of X chromosome equal to 2 and the number of autosomes is equal to 2, then this will be equivalent to 1. Now see this. Which is this type of a Drosophila? This is a type of Drosophila in which two sets of autosomes are present plus XX is present. So when the sex index ratio equal to 1, the Drosophila must become a female. Okay. When the sex index ratio is equivalent to 1, equivalent to half, when it will become equivalent to half, when the Drosophila will be present in the form of two autosomes plus an XY chromosome. Okay, then it would be present in the form of a male. XY means one is X, another one is Y. So the number of X chromosome is just one. Number of set of autosomes is 2. So 1 divided by 2 equals to 0.5. So when the sex index ratio is 0.5, then the organism becomes male. When the sex index ratio is 1, then the same organism is going to become the female. So this is this is the sex index ratio that is the most important one. Okay, so we must know how this sex index ratio is being tabulated and how it is calculated. So if the sex index ratio comes in between 1 to 0.5, the Drosophila will become a hybrid Drosophila. It will become an intersex. Okay, so just writing it out. If sex index ratio becomes greater than 0.5 and becomes less than 1, then Drosophila will become Drosophila will be Intersex. Intersex Drosophila. Okay. Greater than 1. When index ratio is greater than 1, then the Drosophila will be referred to as meta female. Meta female. Meta female or the super Drosophila. Okay. Next, when the sex index ratio is less than, when the sex index ratio is less than 0.5, then it would be referred to as sterile male. Sterile male. Okay. Next, then how does this pattern of expression takes place? Number of X chromosome towards to the ratio of number of X chromosome to the ratio of set of autosomes describes the expression of a gene that is referred to as Sxl gene. Then the next gene that is present is referred to as the Tra gene. Sxl gene splicing will influence another set of gene, and that is referred to as the Tra gene. The Tra gene will influence the Dsx gene, and the male and female flight development is going to take place. Now, how does this entire process is being regulated? Please have a look here. See, it is very beautifully described here, as well as in your book it has been clearly described that alternate splicing take place. Either alternate splicing take place or normal splicing takes place. See, if the sex index ratio, if the sex index ratio is equivalent to 0.5, when the SIR, sex index ratio, is equivalent to half or 0.5, then normal splicing takes place. Normal splicing occurs when the sex index ratio equal to 1. When the sex index ratio is equivalent to 1, then what is going to happen? Then alternate splicing occurs. When alternate splicing occurs, then it clearly means that the Sxl here will lead to the formation of the female. How? This has been normally spliced. Now, the next thing is this Sxl, which was being spliced in the case of female only, will influence the expression of the next protein that is referred to as Tra. Tra is only being synthesized when there is an alternate splicing. Tra protein is only synthesized. Tra only synthesized in female, synthesized in female Drosophila, in female Drosophila during alternate splicing, during alternate splicing mediated by Sxl. Sxl. So it is the Sxl that would be mediating this particular splicing to take place. Next, now, after the Tra would be produced, Tra would produce another type of an splicing in the case of the disc gene. So see, this is what sir has described here. The alternate splicing, normal splicing or alternate splicing. You look here, normal splicing male, alternate splicing female, normal splicing male, alternate splicing Tra protein. Okay, Tra protein will act upon Dsx. In the case of Dsx, see, if the male has to be produced, A and C exons are ligated together. If the female has to be produced, then A and B exons are going to be ligated together. Okay, so female specific Dsx protein is going to be biosynthesized. So here the Sxl gene was having normal splicing or alternate. Okay, normal splicing. Normal splicing will lead to the formation of a male. Alternate splicing, alternate splicing will lead to the formation of a female. Okay, Tra, the same thing happens here. Normal splicing male, alternate splicing female. Normal splicing would be male, alternate splicing will be female. Splicing, and this alternate splicing will be mediated by the Sxl protein. Okay, sorry, might not be visible for you all. Just writing it out. Sxl. This is Sxl. Now the next one is referred to as Dsx protein, which is involved. Now Dsx gene is involved, and here the gene is going to initiate the process of splicing. Here Dsx can be processed into two different types to form A with C or A gene with B gene. It will lead to the formation of male, it will lead to the formation of female. See this, AC is male and AB is female. This is what I have described here. You can note it down, and this is how alternate splicing takes place in the case of Drosophila melanogaster. Okay, is this clear in your mind? Okay, so this is how splicing procedure takes place in the case of a higher eukaryotic organism. I do hope that you would have understood this out. This is all over for today. From tomorrow, we would be talking about the gene, the trans-splicing, and the self-splicing group 1 intron, then the site-specific modification and the insertion and deletion to finish this entire process of splicing. So this would be all completed tomorrow. So that's all for today. Hope you enjoyed the lecture. I would be posting it up, and you can also look at the recorded version for the same. Thank you. Good